Welcome, dear students, to another topic in our Gulel series, that is locomotion and movement. Okay, as you already know, there's a difference between movement and locomotion, where we tell you that every locomotion is movement, but every movement is not locomotion. क्योंकि movement को हम define करते हैं change in the position और posture. जैसे कि ये hand का movement है. But when the organism moves from one place to another place, it is called as locomotion. So all locomotions are movement, but all movements are not locomotion. In the living world, we observe different types of movements. Like if you look at the lower organisms, we see amoeboid movement. Amoeboid movement is with the help of pseudopodia, that is seen in amoeba. Even in human beings, our blood has got white blood cells. Then our tissues have got macrophages. ये लोग भी amoeboid movement दिखाते हैं. Then the second type of movement is the ciliary movement. And this ciliary movement is brought about with the help of cilia. And these cilia are the small hair-like projections. And these cilia move like oars. एक घोड़ी के चप्पू जैसे. Then there is flagellar movement, where the movement occurs with the help of flagella. Like we have seen, sperm has got a tail, and the tail shows flagellar movement. Similarly, many protozoans like dinoflagellates, euglenoids show the presence of a flagella that helps in the flagellar movement. And the topic of our discussion is going to be the movement that occurs due to the contraction and relaxation of muscles, and that is called as the muscular movement. So human beings show movement and locomotion, and we do that with the help of our muscles. Locomotion in kingdom Animalia is with the help of different parts. Like in the case of Hydra, it is with the help of specialized structures, which are called as the tentacles. In the case of amoeba, it is with the help of pseudopodia. And हम इंसानों में, it is with the help of the limbs. यहाँ पे हम बात कर रहे हैं hind limbs के बारे में. और जब डिस्कशन होता है मूवमेंट और लोकोमोशन के बारे में, तो तीन चीजें यहाँ पे डिस्कस होती है. एक, we talk about the muscles, the joints, and the bones. And as you know from our lectures, we say that the bones are the levers, joints is the fulcrum, and muscles provide the necessary effort. तो इमेजिन करिए आप एक सीसॉ को, तो ये जो है वो आपका लेवर है, जहां पे मूवमेंट होने वाला है वो आपका फलक्रम है. मैंने यहाँ पे एक हैवी ऑब्जेक्ट रख दिया, वो एफर्ट है, और इस साइड की तरफ उसका मूवमेंट हुआ. तो सिमिलरली ह्यूमन बॉडी के अंदर मूवमेंट एंड लोकोमोशन दिखाने के लिए आपको लेवर्स की जरूरत है, जो बोन्स है. एफर्ट लगाएंगे मसल्स, और जो पॉइंट पे मूवमेंट होगा दैट इज गोइंग टू बी योर फलक्रम. सो अगर ये मेरा, दिस इज माय हैंड, राइट स्टूडेंट्स, दिस इज द muscle bicep that will provide the necessary effort for the lever, the bones, to move at the point which is joint, that is called as the fulcrum. So that is how you're supposed to understand the use of the muscles, the joints, and the bones in the human body. Talking about the muscles, we know that muscles are of three types, and we have studied them in detail in our previous chapters also. That is, the types of muscles include skeletal muscles, cardiac muscles, and smooth muscles. So here we need not go into the details as we have covered them in our other chapters. But just to quickly revise कि ये टॉपिक डील करेगा स्केलेटल मसल्स के साथ. एक मसल के सिंगल यूनिट सेल को हम लोग बोलते हैं मसल फाइबर. And in the case of skeletal muscles, which are also called as striated muscles, these are unbranched, they are cylindrical, they are non-tapering, and they are multinucleated. They show the presence of peripheral nuclei, and it's a condition which is multinucleate, and it is called as the syncytium. They are voluntary muscles. They are under the control of our central nervous system. So the skeletal muscles, as we have studied previously, they show the presence of the muscle cell, which is the muscle fiber, whose membrane is called as the sarcolemma. The cytoplasm is called as the sarcoplasm, and it shows the presence of multinucleate condition. And this multinucleate condition is called as the syncytium. The sarcoplasm shows the presence of abundant mitochondria. It shows the presence of lot of glycogen reserves. It also shows the presence of a special endoplasmic reticulum that stores calcium, and that is called as the sarcoplasmic reticulum. So there is abundant mitochondria and glycogen, which will provide the fuel for muscular movements. Calcium stored in the sarcoplasmic reticulum, as we'll see further, it will help in muscular contraction. Inside the muscle fiber, there are proteinaceous thread-like structures. This proteinaceous thread-like structures is called as the myofibril. All these myofibrils are parallelly arranged like a bunch of pencils, like this bunch of pencils, and it gives them a striated appearance. Like if I imagine hundreds of zebras standing like a wall, their black and white stripes will result in the formation of dark and light bands throughout their bodies. So if we look at a myofibril under a microscope, we see the presence of alternating light bands and dark bands. And these alternating light bands and dark bands have been taught to you in our animal tissues part. It has alternating light bands and dark bands, but I'll just revise it. The dark bands are called as the A bands. The light bands are called as the I bands. The dark bands show the presence of a light zone, which is called as the H zone. hence S zone. The H zone shows the presence of a M line. The light band shows the presence of a dark line, which is called as the Z line. The distance between two successive Z lines is called as a sarcomere. The sarcomere shows the presence of a full dark band and two half light bands on the either sides. So one sarcomere shows the presence of a full dark band and two half light bands on the either side. If we look at the sarcomere under the microscope, then we'll say that the sarcomere is made up of two types of myofilaments, the thin myofilaments and the thick myofilaments. The thin myofilaments are called actin filaments, and the thick myofilaments are called the myosin filaments. The actin filaments are made up of actin, troponin, and tropomyosin, whereas the myosin thick filaments are made up of monomeric units, which are called as meromyosin. तो सार्कोमियर के पास एक फुल डार्क बैंड है और दो हाफ लाइट बैंड्स है ऑन दी ईदर साइड्स. आपको रिकॉल करना है तो हम ऐसे दिखाते थे उसको कि एक फुल डार्क बैंड है और दो हाफ लाइट बैंड्स है ऑन दी आइदर साइड. तो ये फुल डार्क बैंड हो गया, ये हाफ लाइट बैंड एक तरफ, एक हाफ लाइट बैंड दूसरी तरफ. ये सेंटर में जेड लाइन है. जेड लाइन की दूसरी तरफ जो लाइट बैंड है, हाफ लाइट बैंड है, वो दूसरे सार्कोमियर का पार्ट. सो अगर मैं इस लाइट बैंड को नाम दूं ए एंड ए डैश और इसको नाम दूं बी एंड बी डैश, then this sarcomere has got full dark band with an edge zone and the M line and A dash and B. और इसी सार्कोमियर को हमने यहाँ पे अभी मैग्नीफाई करके दिखाया हुआ है. So that is why I've been repeating, and you're supposed to learn that part, that one sarcomere shows the presence of a full dark band and two half light bands on the either sides. The sarcomere consists of the thick filaments. So do you see this part that is shown in the red? That is the myosin filament, thick filament. And these are the thin filaments. This is the, repeat in your mind, this is the Z line. This is the Z line. Now, remember, when there is a thick filament, which is called as the myosin filament, only thick filament, light will pass through it. When there is only a thin filament, that is actin filament, light will pass through it. But when there is a thick and a thin filament, which is overlapping, light will not pass through it. Now, so if you look at this representation of the sarcomere, you will observe that this is the region where thick and the thin filaments are overlapping. So that will give you the dark band. But in the center of the dark band, there are thick filaments which are not overlapped by the thin filaments. So here light will pass and that will give you the H zone or the Hense's zone. And here we have only the thin filaments जिसके through light pass होगा. It will have only the actin filaments. So as a result of which you will have the half light band. Okay. So आपको डार्क बैंड क्यों मिला? कि वो ऐसा रीजन है जहां पे थिन और थिक फिलामेंट्स ओवरलैप कर गए. फिर डार्क बैंड के सेंटर में एच जोन क्यों मिला? क्योंकि वो एक ऐसा रीजन है जहां पे थिन फिलामेंट्स ने थिक फिलामेंट्स को ओवरलैप नहीं किया. साइड में आपको दो हाफ लाइट बैंड्स क्यों मिले? क्योंकि वहां पे सिर्फ आपको मिलता है थिन फिलामेंट्स, वहां पे थिक फिलामेंट्स नहीं मिलते. राइट नाउ, so after understanding this, let us recap what is a H zone. So I said, let us recap the H zone. So now this is a muscle or the sarcomere in the resting state. Okay. And in a resting state, we find that the edges of the thin filament, the edges of the thin filament partially overlap the free ends of the thick filaments. ये जो thin filaments की edges हैं, वो thick filament के free ends को partially overlap करती है, leaving the central part not overlapped. And that central part of the thick filaments, which is not overlapped by the thin filaments, is called as the H zone. Now, we'll further discuss the thick and the thin filaments. The thick filament or the thick myofilament is the myosin filament. It is made up of monomeric units, which is called as meromyosin. And the meromyosin is made up of 6 polypeptide chains, out of which 2 of them are heavy and 4 of them are light. कितने polypeptide chains है? 6. उसमें से 2 heavy chains है और 4 light chain है. एक heavy chain के पास हेड है, ग्लोब्यूलर हेड है और टेल है. ऐसे एक ग्लोब्यूलर हेड और एक टेल, एक और ग्लोब्यूलर हेड और एक टेल मिलके हो गया आपके 2 हेवी चेन्स. और एक हेड के पास 2 लाइट चेन्स आ गई. तो टोटल लाइट चेन्स कितनी आ गई? 4. इसलिए मैंने कहा 6 पॉलीपेप्टाइड चेन्स है, जिसमें से 2 ऑफ देम आर हैवी and 4 of them are light. Now, further discussion is that meromyosin, we are clear, has got these हेवी चेन्स एंड लाइट चेन्स, ये सब मिला के जब एक मीरोमायोसिन मॉलिक्यूल बनता है तो द ग्लोबुलर हेड along with a short arm is called as the HMM, or the heavy myromyosin, and then only the tail part is called as the LMM, that is the light myromyosin. So we are clear with this part, that each myromyosin has got two important parts. These two important parts are the HMM and LMM. The HMM includes the head and the short arm. Head and the short arm. Now, this head and the short arm, the head and the short arm at regular intervals will protrude out of the myosin, and it will result in the formation of the cross arm. So the HMM component, that is the head and the short arm, project outwards from the myromyosin and results in the formation of the cross arm. इसको हम ऐसे समझ सकते हैं कि बहुत सारे मीरोमायोसिन ये साथ में आए and they resulted in the formation of the myosin, and हर एक मीरोमायोसिन का HMM component, which includes the short arm and the head, at regular intervals will protrude out, and it will result in the formation of the cross arm. और यही क्रॉस आर्म हम देखेंगे आगे चल के क्रॉस ब्रिज बनाएगा. तो अगर हम ऐसा इमेजिन करते हैं कि मेरा हेड और मेरी नेक, ये हेड है और ये नेक है शॉर्ट आर्म, ओके. बाकी का पूरा पार्ट LMM है. हेड एंड ए शॉर्ट आर्म मिलके हम बोलेंगे कि ये HMM वाला कंपोनेंट है, और ये ऐसे प्रोट्रूड आउट करता है. जैसे बच्चे जब स्कूल बस में बैठे रहते हैं, उनकी पूरी बॉडी बस के अंदर है, वो LMM वाला पार्ट है, लेकिन उनकी नेक और उनका हेड जो HMM वाला पार्ट है, वो ऐसे खिड़की के बाहर निकलता है. तो वो जो प्रोट्रूड आउट वाला पार्ट है, उसको हम कहेंगे कि वो क्रॉस आर्म है, और यही क्रॉस आर्म आगे चल के बनाएगा क्रॉस ब्रिज. देन ये समझने के बाद हम एक और चीज समझेंगे यहाँ पे, that the head shows the presence of some important sites, like it shows the presence of an ATP binding sites. It shows the presence of ATP binding sites, where the ATP molecule will bind to the head of the myosin. Similarly, on the myosin, there are actin binding sites. Similarly, third thing, this head has an ATPase activity. This head has an ATPase activity, where it can hydrolyze the ATP into ADP and inorganic phosphate and cause the release of energy, as a result of which the myosin head will get energized. So it shows the presence of actin binding site, जिसके मदद से मायोसिन का हेड बाइंड करेगा एक्टिन को. इट हैज गॉट एटीपी बाइंडिंग साइट्स, जहां पे एटीपी बाइंड करेंगे मसल कॉन्ट्रैक्शन के टाइम पे. और फिर मायोसिन के हेड के पास एक तीसरा प्रॉपर्टी आप ध्यान रखेंगे कि वो खुद एक एटीपीएज एक्टिविटी दिखाता है, जो एटीपी को हाइड्रोलाइज करके एनर्जी रिलीज करवा सकते हैं. तीन वेरी इंपॉर्टेंट चीज है, आप सबको याद रखना है. सो एक क्विक रिकैप. The sarcomere is made up of thick myofilaments. The thick myofilaments are made up of myosin. The myosin is made up of monomeric unit, which is called as the meromyosin. And the meromyosin is made up of two components, the HMM and the LMM. The LMM has only the tail part. The HMM has a globular head along with the small polypeptide chains and a small short arm. The head and the short arm at regular intervals will protrude out, resulting in the formation of the cross arm. Later on, the cross arm will result in the formation of the cross bridge. The head shows the presence of an ATP binding site. It shows the presence of actin binding sites, and it shows the presence of an ATPase activity where the head can hydrolyze ATP and it will cause the release of energy. That is about your thick myofilament. And now we discuss the thin myofilament. The thin myofilament is made up of three components here: actin, troponin, and tropomyosin. Actin, troponin, and the tropomyosin. Actin के दो फॉर्म्स होते हैं. एक होता है ऐसा ग्लोबुलर फॉर्म. यही ग्लोबुलर फॉर्म साथ में आता है Mg प्लस के प्रेजेंस में, एंड दे रिजल्ट इन द फॉर्मेशन ऑफ द एफ एक्टिन. तो ये जो मैंने गोले 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 के साथ में बनाए हैं, ये ग्लोबुलर साथ में आके एक एफ एक्टिन बनाए, और एक गोले 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 ग्लोबुलर साथ में आए और एक दूसरा एफ एक्टिन बना. तो ऐसे दो एफ एक्टिन होंगे एक थिन फिलामेंट में. Similarly, the second component is, you must be seeing in the diagram कि ये F actin है, and along the F actin we see the presence of helically two filaments of helically wound protein along the F actin, जिसको कहते हैं ट्रोपोमायोसिन. ओके, सो ये ट्रोपोमायोसिन हो गया, which consists of two filaments along the length of the actin. And the third component is a protein called troponin. And the troponin is made up of three subunits. The troponin is made up of three subunits. We don't need to remember the name, but now remember the job of troponin. इस एक्टिन के यहां पे मायोसिन बाइंडिंग साइट्स है. जैसे मायोसिन के पास थे एक्टिन बाइंडिंग साइट्स, एक्टिन के पास भी है मायोसिन बाइंडिंग साइट्स. When a muscle is in a resting state, when a muscle is in a resting state, the troponin masks the myosin binding site on actin. मतलब ट्रोपोनिन की वजह से रेस्टिंग स्टेट में मायोसिन आके एक्टिन को बाइंड नहीं कर सकता, क्योंकि ट्रोपोनिन उसको मास्क करके रखता है. सो ये एक्टिन है, ये मायोसिन है. अब in a resting state can the myosin bind to actin? No, because the binding sites are masked by troponin. तो आपको सबसे पहले ट्रोपोनिन को हटाना पड़ेगा, अनमास्क करना पड़ेगा, तब जाके मायोसिन बाइंड करेगा एक्टिन के साथ. Okay, so let us discuss the sliding filament theory. Now we will continue. The sliding filament theory tries to explain the mechanism of muscle contraction. Okay. Now, how do we understand the sliding filament theory? So, in a point-wise manner, for the muscle contraction to begin, these are voluntary muscles. So, the signal for their contraction will come from the central nervous system with the help of a motor neuron. And these motor neurons, they come close to the sarcolemma. Now, the axon knob and the sarcolemma together is called as the NMJ, or the neuromuscular junction. So, a motor signal will come via the motor neuron of the central nervous system. It will reach the neuromuscular junction. After it reaches the neuromuscular junction, the axon knob will release acetylcholine. The acetylcholine will bind to the acetylcholine receptors which are there on the sarcolemma. As a result of it, the sarcolemma will get depolarized. Due to the depolarization of the sarcolemma, the calcium gates of the sarcoplasmic reticulum will open. Calcium का content in the sarcoplasm will increase. Calcium will bind to troponin and it will unmask the myosin binding sites on actin. As a result of which, the head of the myosin is now ready to bind with the actin. The head of the myosin, as I have already told you, it has got an ATPase activity. So the head, which has got an ATPase activity, will hydrolyze ATP into ADP and IP. As a result of which the head will get energized. That is the first step. And ADP and phosphate is still attached to the head. Now the head, the cross arm, will bind to the actin binding sites. The head, the cross arm will bind to the myosin binding sites on actin. And that will result in the formation of a cross bridge. Now the cross bridge will rotate inwards, causing a power stroke. Actin के filaments Z लाइन से अटैच्ड. अब पावर स्ट्रोक के दौरान एक्टिन के फिलामेंट्स मायोसिन के ऊपर स्लाइड करेंगे और ये एक्टिन Z लाइन से अटैच्ड है. So even the Z line will start moving inwards. So the length of the sarcomere will decrease and the muscle will undergo contraction. So here, these are the two sarcomeres which I have shown, right? These are the I bands. Half and half together form full I band. This is the A band. This is a muscle sarcomere in a relaxed state. Now during power stroke, as the myosin head will rotate inwards, the cross bridge which is holding the actin will cause the actin to slide over myosin. As the actin will slide inside, it is attached to the Z lines, so Z lines भी अंदर की तरफ आएंगे. As a result of which we see in a relaxed state and in a contracted state, compare the position of the Z lines. The Z lines have moved inward. The I band has disappeared. Even the H zone has disappeared. But we can still see the A band. That is why during power stroke, as the actin slide over myosin, the Z lines are pulled inwards, I band and H zone disappears, and the sarcomere undergoes contraction and the muscle will undergo contraction. अब मसल को रिलैक्स करने के लिए the ATP will bind to the myosin head, and as the ATP will bind to the myosin head will leave the actin filament and the muscle will undergo relaxation. So in the last step, ATP मॉलिक्यूल आके बाइंड करेगा मायोसिन के हेड को. जैसे ही ATP मॉलिक्यूल बाइंड करता है मायोसिन के हेड को, मायोसिन का हेड एक्टिन फिलामेंट को छोड़ देता है, जिसके वजह से एक्टिन जो है अपने ओरिजिनल पोजीशन पे चला जाता है and it will undergo relaxation. So that is the muscle contraction cycle. The types of muscles. If the sarcoplasm shows the presence of a lot of myoglobin, it is reddish in color. So it is a red muscle fiber. It has got more blood capillaries and it undergoes aerobic oxidation. It can undergo slow rate of contraction for a long period of time. इसके कंपैरिजन में होते हैं व्हाइट मसल फाइबर्स, इनका डायमीटर बड़ा होता है लेकिन उनके अंदर मायोग्लोबिन नहीं होता या वेरी लेस होता है, तो दे हैव टू बी रिलाई अपॉन एनारोबिक रेस्पिरेशन फॉर देयर कॉन्ट्रैक्शन. Okay, with respect to the muscles, this is what you're supposed to remember. Finally, remember that muscles are the efforts which will give the necessary force for the muscle to undergo contraction and relaxation. The muscles are made up of muscle fibers. These muscle fibers form fascicles, and the fascicles are joined together by fascia. This last point we have discussed in animal tissues. So that's it for muscle part. Now we can begin the discussion of the skeletal system.